Dear learners, I am Ramya, working as Assistant Professor in the Department of Biotechnology, St.Josephs College of Arts and Science for Women, Hosur. We are going to learn about preservation of cultures, how the culture is preserved after the subculture, and it is maintained for extended periods in a viable condition without any genetic changes is called as preservation. During preservation, most important factor is the preserved culture should stop the microbial growth and at least to lower the growth rate. Due to this, toxic chemicals are not accumulated and hence viability of microorganisms will not be affected. The techniques mainly used for academics, research purpose, fermentation processes and in biotechnology field. There are mainly 6 methods for this preservation. Agar's land culture covered with oil, saline suspension, preservation at very low temperature, preservation of cultures by drying in vacuum, lyophilization technique or freeze drying. We are going to see in detail. Agar slant culture. Agar slants are prepared in vitro. After inoculation, slants are incubated for a period of 24 hours, then stored in refrigerator. This requires periodic transfer after 6 months. Once this agar slant culture, pure culture is maintained, it has to be transferred after 6 months, otherwise it will be contaminated. Next is agar slant culture covered with oil. The agar slants are incubated after inoculation until profuse growth appears. Then it will be covered with mineral oil at a depth of 1 centimeter on the tip of slanted surface. Transfers are made by removing a loop of growth. They touch the loop to the glass surface to drain off the excess oil in the medium and then preserving the initial block culture. Next method is saline suspension. High concentration of sodium chloride is used as inhibitor of bacterial growth. Bacteria are suspended in 1% salt solution in screw cap tubes to prevent evaporation. The tubes are stored at room temperature and transfers are made on agar slants. Preservation of culture at very low temperature. The organisms are suspended in nutrient broth containing 15% glycerol or in skimmed milk containing 7.5% glucose. The suspensions are frozen and stored at 15 degrees Celsius to 30 degrees Celsius. This ready availability of pure culture is maintained in liquid nitrogen, that is minus 196 degrees Celsius, has provided another means of preservation of stock cultures. In this procedure, the cultures are frozen with protective agent like glycerol or dimethyl sulfoxide in a sealed ampules. The frozen cultures are kept in liquid nitrogen flask. Preservation of pure culture by drying in vacuum. The organisms are completely dried over calcium chloride in a vacuum, then stored in the refrigerator. The organism survives longer than when air dried. If we dry in calcium chloride, the life of a microorganism will be there compared to air drying. Lyophilization or freeze drying. The microbial suspension is placed in small vials. A thin film is frozen over the inside surface of the vial by rotating it in a mixture of dry ice or alcohol or acetone at a temperature of minus 78 degrees Celsius. The vials are connected to high vacuum line. This dries the organisms while still frozen. Finally, the ampules are sealed off in a vacuum with a small flame. These cultures can be stored for several years at 4 degrees Celsius. To revive microbial cultures, it is merely necessary to break up the vial aseptically, to which suitable sterile medium is added. After incubation, growth appears which allow them to further transfer. The process permits the maintenance of a large number of cultures without variation in the characteristics of cultures, which generally reduces the danger of contamination. These six methods are useful in preserving the culture. My question for today's learning is, which goes for short method of preservation and easily it can be transported. Thank you, dear learners.